Hide. Okay, right. So instead of doing things the other way and you get the quiz before the lecture, I thought this was a good idea to do the lecture first. So, okay, so we're going to talk about saturation under exposure, bit depth, and image display. That is a full program. And we have touched upon the saturation and a tiny bit about underexposure so far. We haven't said anything about bit depth. But it's possible that you guys have seen already something about bit depth. So we're going to make everything quite clear, hopefully, now. Yes, now. Okay, so we see in this image, there is an overlay and 3 channels, 1, 2, and 3. And we can wonder what is wrong with this image. So to start with, we could think that maybe channel 1 and channel 3 are very dim, and that's not very good. But really the problem lies in channel 2. And indeed, it is saturated and not the others. So how do I know that it's saturated? I will show you. So what does that mean, that it's saturated? Well, if I put if I put my mouse across this area, so right now I have applied a mask that allows me to see the saturation, and it's color-coded in red. So everything that is red in this image is saturated. So if I put my mouse above the one of the places that are saturated, what I can see under the image, and again, this is something that you can do on all software. If you move your mouse across the image, somewhere in the software, you should be able to see the XY position where your mouse is, and also the intensity at that point, and the intensity of each channel. So in this particular case, we have 3 channels. So this is the XY intensity, and these are the 3 channels coming from the 3 different channels, the 3 intensities. And what we can see in here is that the green channel at the place where I have my my mouse has the intensity of 4095, which is a number that maybe some of you may recognize. So what does saturation look like? If we look a bit closely at this image, we have all these pixels that are in red have the same intensity, 4095, which if you think about a software, it's very unlikely that this is like a sample. It's very unlikely that it has so many areas with exactly the same intensity. So if we remove the saturation mask, this is what the image looks like. And so when you look at your images, most of the time you look at them this way, and this is what you should look suspiciously at. We have some areas that look solid with one color, and there are no details at all in these areas. And so this is an artifact of imaging, and the red pixels or solid white pixels are saturated. So what does that mean that they are saturated? If we have, this is a small fluorescent bead, and this one is well exposed, and this one is saturated. And we see this because we've applied the saturation mask. And when we we draw a line across the bead and we look at the intensity profile, what we can see in this well-exposed bead is that actually there is a texture in the bead. We can see some details. The intensity changes a little bit. So there is some sort of, we can see some texture in here. And this is reflected in the intensity profile where we have some variation here at the top, where the line crosses the bead, and then it goes back down because we're in the background. If we now look at the saturated bead and we do the same thing, the histogram has a flat top. So there are absolutely no details recorded in this area, and this is the reason why when we look without the saturation mask, everything looks solid white, and we see no details at all. And all these pixels here that are color-coded in red have the maximum possible intensity in the image, which is 4095, and we will see why. So the intensity information recorded in these pixels is incorrect. It is not the correct intensity. There are absolutely no details. What we need to do is go back to microscope, lower the exposure time, for example, the gain, the illumination power, the scan speed, lower something so that there is less light on this sample, and re-image. So you have to be also aware that when you do Z-stack, you may set your settings onto one Z plane and everything looks good. You have no saturation, but it is possible that you, when you go to a different plane, it is saturated. So when you do a Z-stack or when you do a time lapse, you have to consider with your best knowledge of the sample and the experiment, what will happen in time. Is the fluorescence going to increase? Then you have to adapt your settings so that you have no saturation at the end, for example. And in Z-stack, as you know, close to the cover slip, mostly we have the brightest sample, but it's not always the case. So it depends on your experiment. So this is something that you have to be aware in Z-stack and time-lapse, what happens in time. So what does underexposure looks like now? Instead of saturation, we also have underexposure. Well, all the pixels in blue. So we have also applied this mask that shows us underexposure and saturation. All these pixels in blue have the intensity of 0, which again, if you think about the sample, it's pretty much not possible. So it's artificial. If you do not have the saturation and underexposure mask, it is much harder for you to see that there is a problem with the images when they are underexposed. So it's this is solid black, but since anyway it's normally quite black, we will not notice until we put the saturation mask. So without the mask, our eyes cannot see, but there are no details in the background at all. So this is also an artifact, and all the blue pixels are underexposed. It's a very nice background, right? But there are no fine intensity details at the cell edges, and everything has been eaten up. And it's easier to detect saturation with our eyes. We see these solid white areas, but it's much harder to see underexposure. So this image is very likely to be rejected by a journal. If I was the reviewer for this paper, I would definitely reject that and say, go back to your microscope and reimage because it cannot be corrected afterwards. So where does saturation and underexposure come from? Well, it comes from the dynamic range and the bit depth. And how can we detect it? We're going to look at so this is the sort of the outline of the little lecture today. How can we detect saturation and underexposure? We have the lookup tables and the histograms, so I will explain that. What are the consequences? So there's no usable information in these areas, and we'll talk about that one. And then how to change the image display correctly. And then we'll talk about brightness, contrast, and gamma. So let's talk about the first one. What does saturation, where does it come from? So the bit range and it was dynamic range and bit depth. Right, so again, if we have a look at the image, and in the Nikon software, it's at the bottom of the image, but in the Zeiss software, it's somewhere else, and you have to figure out where in the software is the bit depth written. It's definitely written somewhere. So, and you will have to find out in one of the portfolio assignments. So, right, this is the bit depth. It says 3 times 12 bits, and it's 3 because we have 3 images. So each image has 12 bits. So what is this? So if we go back to what a good sensor is that we have seen before, we have a number of steps. From the incoming photons all the way to getting a final image. We have some detection of the photons, amplification, and then conversion to a digital signal. And the bit depth and the dynamic range concern this last step of reading the voltage and converting the signal to a digital signal. The dynamic range of the sensor is the ability of the sensor to read dark and bright areas with intensity details and without saturating. This is a capacity of the sensor. So when you buy your sensor, it already has a maximum dynamic range. And once you have bought it, you cannot do anything about it. So this is part of the sensor specifications. So you cannot so it cannot be chosen in the software, but you can influence the dynamic range in the software by changing the bit depth, for example, by changing the readout rate. This will influence the dynamic range. It cannot be changed after acquisition. So the dynamic range you have, that's what you get. So on camera-based systems, we have a high quantum efficiency. So I remind you, quantum efficiency is the percentage of incoming photons that are going to be detected by the camera or the sensor is the quantum efficiency. In camera-based systems is like over 80%, so it's very high. And also the exposure time that we use with a camera-based system is in milliseconds. And I know that some of you image in seconds, but that's not normal. So normally it's milliseconds, maybe 10, 20, 200, something like this. These are sort of the normal exposure times that we use. So it's quite long, if you want to say. And also, the way cameras work, and we will see this in the camera workshop a bit later, the way cameras work is that they accumulate electrons before the chip is actually read and emptied. So there's an accumulation. So there is a high risk there in cameras for saturation. So the cameras require a high dynamic range because of this accumulation and the fact that we have long exposure time, etc. And detector-based systems, that the single point confocals, they are very slow, but they stay very short time on each pixel. So they have a low quantum efficiency, so not so many photons are detected. They stay on only microseconds on each pixel. And also the electrons are read on the fly, so there is no accumulation. So in general, in detector-based systems, there is less need for high dynamic range. Usually they work with low dynamic range quite okay. So what is then, we have seen that the dynamic range is the one that sets the bottom floor and the top of our dissection. So this is the one that determines if we're going to saturate or not. So how can we affect the dynamic range since we cannot do anything and there's no setting in the software? We can change the bit depth in the image, and that will affect the dynamic range. So what is the bit depth? And that is a specification of the image now, not of the sensor. The bit depth is the number of intensity steps between dimmest, which would be completely black, and the brightest, which would be completely white. So it's the number of steps between the possible intensities in the image. So that is the maximum possible, and this is the minimum possible. It does not mean that this is actually the minimum and the maximum in the image. It's just the maximum we can get. So this can be set in the software, and that's true for both detectors and camera systems. And it can be changed also after acquisition. So in this image, we have an 8-bit image. What does 8-bit means? It just means 2 to the power of 8. And therefore, an 8-bit image has 256 steps or intensity values, gray levels. All these mean the same thing. It's called sometimes this way. And the first step is always 0. And so the last step is, guess, 256 minus 1. So because the first one is 0, so we have 255 at the maximum for an 8-bit image. And some of you will recognize this value as familiar because when you saturate, this is what you get as an intensity. And then anything in between is gray and has an intermediate value. So here we're looking at a 16-bit image, and you can see that it has a lot more gray levels possible. The 0 is always the first one, and so 16 bits, so 2 to the power of 16 means 65,536 levels, and so we have that minus 1 for the highest, the completely white. And you can see that we have like very smooth gradient of intensity. So a high bit depth image is very good for you to see the fine gradation of the intensity. Whereas a low bit depth image, you will just get like big steps basically. So it's either one color or the next, but you have no fine details in the intensity. Our computer screens can only read 8 bits, and our human eyes can only read 7 bits. So we're not very, very good with our eyes, and this is why I keep saying that do not believe your eyes. Please look at the data. So explore your image with the mouse and see the intensities instead of believing your eyes. The image contains a lot of very dim information that our eyes does not see, and this is the reason why we changed the display to be able to explore the image. So do not just set everything based on your eyes. Okay. So basically, when we have a 16-bit image, anyway, this is a 16-bit image, but it's only displayed in 8 bits by my computer screen. And my I can hardly see anything at all. Right, so just to do a little quick round in Zen, in the Zeiss software, this is where you find the 8 bits. It's just, the bit depth is just above the scan area, and you can change here. And in Zen, the default bit depth on single point confocal is 8 bits, and you can change it to 16 bits. If you if your sample is in such a way that you quite easily saturate it. And we'll talk about when this happens. In Fiji, you can also find the bit depth, and it's in here under image type, and you have the bit depth, and this is where you can also change the bit depth. So why would you change the bit depth? Because non-scientific software like PowerPoint, for example, cannot read an image that is in a higher bit depth than 8. So when you have such an image, you have to convert it to 8 bits. Of course, you don't do it on the original one, you do it on a copy, and then you can put it in your PowerPoint presentation. And if you save it in JPEG, you get automatically, of course, not the original one, right? You save a copy in JPEG, it automatically goes to 8-bit. So only change the bit depth on a copy of the original, keep the original intact. This is very important, and that can be used also if you want to reduce the downsize, the size of the file to analyze the images easier. But then in that case, you should really acquire them in 8 bits to start with. So you can change from low, so changing from a low to a higher. So if you have an 8-bit image and you go to 16, there's no point because you're not going to create data. So it just makes your image bigger, your file bigger, but it doesn't create anything. There's no advantage. So just don't go upwards. I wrote quiz 2, but we're going to do them afterwards. I forgot to remove that. So this is the bit depth. And we can see here in this table the different bit depths and the levels of gray. So 2 to the power of the bit depth, that's the level. So you can recognize now 12 bits where we have 4096 levels of gray. The first one is 0 and the maximum is 4095. And this is the reason in the images that I've shown you, all the red areas had this intensity of 4095. And these files, only the 8-bit image can be read by any image software, and the others require scientific software, or otherwise you need to downgrade, let's say, a copy of the image to 8 bits. So the advantages of the high bit depth is that first you have access to the full dynamic range of the sensor. So the complete things, the maximum that the sensor can give you between the minimum and the maximum. So the full range basically of intensities that the sensor can detect. You will get fine intensity details in both the bright and the dim areas without saturating. So there is a much lower risk for saturation when you use a high bit depth compared to a low bit depth. The file size can be very different. So you can have even 11 and 14 on some cameras. But everything that does not have 8 is basically all the files have the same size. So 11, 12, 14, 16, all these will result in the same file size because computers can only deal with 8 or 16. So they pretend these are 16. And then, that's interesting. And then the when we have a high bit depth, we have a much finer grading of intensity, and therefore all the edge definition is better, and it might be easier to segment the crowded objects, for example, where when the two objects are close to each other with a high bit depth, you might not see the from one object to another, but with a high bit depth, you will be able to see all these very fine details in intensity. So that might help in the image analysis as well. Some applications for high bit depth, if you have a sample that is very dim in some areas or very bright in others, and then you try to image both of them, you're interested in details in both of them, then you should have a high bit depth, because otherwise if you set the macroscope to be able to detect clearly the dim areas, you're going to saturate the bright ones. And if you set for the bright ones, you're not going to see the dim ones. So use about high bit depth in this case. And another application is if you have a change in intensity over time. So we start with things that are not fluorescent or hardly fluorescent, and over time they become very fluorescent. So this is also a typical application for high bit depth. And so why is it that anyone would want to use a low bit depth then, since the high bit depth is so nice? Well, maybe your scientific question does not require to acquire very fine intensity details. And also, if your sample is dim, you have no risk to saturate the detector, so you do not need a high bit depth. If you use a low bit depth, the images are not so heavy, so it's much easier to go fast. So you get access to the highest speed of the detector when you use a lower bit depth. And then when you save space, clearly the file size is different. So 8 bits is half of the others. And the 8-bit images are a lot less resource demanding when you do analysis. So with the 16-bit images, if you have a multi-dimension image, it might be difficult to for your computer to analyze it. But if they are in 8 bits, it will work much better. So you can say that, and also, of course, the 8-bit images can be directly inserted into PowerPoint presentation, for example. Otherwise, the high-bit images, you have to convert them first. So, and but one thing that is important to remember is that if you want to compare images, they must have the same bit depth. They must be acquired in the same way. Otherwise, they are not comparable. So basically, in general, you're going to go for a low bit depth, 8 bits, and then if your experiment requires, then you move to a higher bit depth. Sometimes in some software, depending on the hardware, 8-bit is not available and only 12-bit is available. But it's the same thing there, where you will have the same file size, but you will be able to go faster with 12-bit compared to 16 bits if you want to image fast. So this is an example of the specification sheet of a camera that you can find online. So you will have to figure out the specifications of your own detector. So this camera is the kinetics camera from Photometrics, and it has 3200 by 3200 pixels, and the camera pixels are 6.5 microns. So now you know exactly what that means. And what we see here in this data sheet is that we have the dark currents with specifies the lower part of the dynamic range, and we also have the full well capacity, which specifies the highest part of the dynamic range. And you can see that, so basically the dynamic range is a ratio between these two. And you can see that when we go very fast, we are not at all getting a high dynamic range. So this is a very low dynamic range from this to this. And but when we go with the high bit depth of 16-bit, then we have a very high, the full dynamic range of the detector. So however, if we look at the frame rate, the maximum frame rate that you can have at these different bit depths, you can see that the 8-bit image you can acquire very, very fast at almost 500 frames images per second, whereas the 16-bit and the 12-bit are much slower. And this is the read noise. So same thing in if you go very fast, very often speed goes against noise. So then you get more noise in these images that are acquired in 8 bits. So this is what we would like. Here we have basically a high dynamic range. Here we have high speed, and here we have a compromise in 12 bits. But all these numbers are different for different detectors. Okay, so we can figure out, depending on our application, which bit depth we want and how bright our sample is. But the low bit depth is some sort of your where you start. So now we're going to talk about how to detect saturation and underexposure. So lookup tables and histograms. So I've shown you already that we have a little button that allows us to see in red the saturated pixels which have the maximum bit depth, and then in blue the pixels that have a 0-bit depth. So I remind you that most sensors acquire images in black and white, and what we have is we add a color to it. So this color is actually called lookup tables, LUT. And yeah, I find it's a little bit funny name, but this is the way everybody calls it, so we call it that way. Lookup tables or lookup LUTs or LUTs or whatever people want to call it are tools to help us see the image. So this is for our eyes, basically, because our eyes are not very good. So it can be, as we have seen before, an artificial color, where the image goes from is black and white, but we're putting it in green. in here. Or we can also change the brightness and contrast. And that's also a lookup table. And then we can change the intensity. We can color code in Z, for example, etc. So we can do all sorts of things with lookup tables. And these are just tools for us to see. They change how, they change how the computer displays the data, but the data is not modified. So that's very useful for us, and we should play with this all the time. So this is available on all software. We have one lookup table that shows you saturation and underexposure. So somewhere in the software, there is something like this. And personally, I think that it is best to keep it on all the time. So in the Nikon software, it's this little button here that shows the blue minimum and red maximum. And when you click on that, the images that are, if you have not displayed them in black and white before, but you should, they become black and white. And also it shows the saturated pixels in red, and that's the maximum bit depth. And it shows the underexposed pixels in blue, and that's the pixels that have an intensity 0. And you can change this in the software if you want to put different colors or something. We have the same thing in other software. These are Zen and Fiji, for example, and where we can first reset the brightness and contrast. In Zen, I find it a little bit, they have changed things a few years ago. Before it was working the same, that it was showing you the saturated and the underexposure. But if you push the brightness in Zen, you anyway get some red now. They have changed it, and I don't think they've made it better. So just reset the brightness and contrast before, although I haven't tried the latest software. Maybe it was a bug and it's been fixed. So you guys can tell me. And then to detect the saturation under exposure. So in Zen, it is called the range indicator. You might have seen it somewhere. And in Fiji, it's called high-low. And so and in Leica, it's called LUT. And here in Fiji, you have a little button called LUTs, and here you can select high-low, and then this is what will happen. So when shall I check underexposure? You can only check the underexposure and the saturation after you've acquired the images, when you're by the microscope. But what you should have is you always should have this underexposure and saturation LUT on when you're imaging. Oh yeah, but I cannot see my channel color. Well, good, because anyway, the images are black and white, and our eyes are not good at all with blue and red. For example, we're much better with green. So always look at the images in black and white to avoid bias when looking where green looks brighter. And then, yeah, all this blue and red in my images prevent me from seeing my signals. Well, yes, great. Then if the acquisition settings were correct, there would be no blue and red. So change the settings. If it bothers you, do something about it, and then it will disappear magically. So now another thing that we can look at to explore the intensities in our sample is the histogram. So again, it's available in all microscopy software, but this is in the Nikon software that we are showing it. So the histogram shows you the number of pixels for each intensity. And what you can see is that you can say, okay, at this intensity, for example, there are about 1,000 pixels that have the intensity of 150 or 160, something like this. Yeah, so it's a histogram. So this image is in bit depth, in 12 bits. And so the minimum is 0 and the maximum is 4095. So this we know now. And so when I look in Nikon software, I can put my mouse here and see the distribution of all the pixels that have the intensity 0. Oops, I can see that a lot of pixels have the intensity 0 in the red and in the blue channel, but nothing in the green channel. So I have underexposure in the blue and the red channel. And then if I look at my mouse here, I can see that in the green channel, lots of pixels are saturated, but not in the blue and red. So there is saturation in my images. And also I can see that most of my photons that I'm collecting are in this part of the range, and the rest of the intensity range is not used. So with the blue and the red channel. So this is not very good. And here with the green, I have quite a good, I'm using the full dynamic range here, and I have quite a good full histogram. So I'm filling up the histogram and stretching. So the green is better than the others, but it's anyway saturated. So we have to do something for all these channels, basically. And so these are the settings, and we will see we will go through all these settings in this typical workflow on how to set a single point confocal this afternoon. But anyway, once I have changed all the settings, I have now a correct histogram where I'm using the whole histogram, but I have no underexposed pixels and no saturated pixels. And if I do not push the brightness and contrast, this is what images looks like. And you can see that it's very different from before. Yes, so make sure that you have no saturation at all and no underexposure at all in the areas of interest. And so in the areas of interest, it means what? If you have, for example, a blood antibody that is here, and that's not what you're interested in, but if you would set the image, the settings in such a way that this one would not be saturated, then you're not going to see anything else. So if you have something like this in your image and it's not in the area of interest, it's fine. What you can do maybe is try to not include it in the image, but that is okay if this type of things are saturated. It's only in the areas of interest you should have no saturation, no underexposure. And again, I just want to point out that the maximum bit depth, so 4095, is not the maximum intensity in the image, and 0 is not the minimum intensity in the image. These are the possible values in the image. If the image is not saturated and not underexposed, we have no values at the minimum and at the maximum. So the maximum intensity is below the maximum bit depth. So the bit depth is just the maximum we could get. So in Fiji, if you want to look for the histogram, you'd go to Analyze and Histogram, and then you get this. And here we can see the minimum and maximum in each of the channels. Okay, we're going to have a tiny break. I'm going to have a look how good I'm doing or not. We're going to have a break now. Sorry, yeah, a little break of 5 minutes. And so we are coming back just shortly before 11.